Okay guys, 3.3.4.2, mass transport in plants. We're just going to talk transpiration in this first part, we'll talk translocation in the second part. So you remember from GCSE that a plant has leaves, it has a stem, it has roots. It's the roots where the water gets in by osmosis and it is pulled up the stem and moves out the leaves through holes in the bottom of the leaf called stomata. This evaporation leads to the water column kind of being dragged up, and we're going to talk about this and how it might be affected. Now, when we looked at biological models, we looked at water, we talked about something called cohesion tension, so look back at that if you need a refresher. But cohesion tension essentially is what allows this process to happen, okay? So what's taking place is that in the stomata, we get, if we drew guard cells down here, and then we have the mesophyll cells, if you remember, and then we have the palisade cells at the top, and we have the waxy cuticle, the upper epidermis. So the xylem vessels are where the water is appearing. So as oxygen is moving out, that's being made in the palisade cells during photosynthesis, and carbon dioxide is moving in, and that's going to be used in the palisade cells, photosynthesis. So long as the stomata are open to allow that gas exchange process to happen, this water has the opportunity to evaporate out of the stomata. So at night when the plant isn't photosynthesizing, these are closed and this prevents this. But as that water molecule moves out, it pulls with it another molecule behind it as a result of cohesive tension. So there are hydrogen bonds between the oxygen atom on one molecule and the hydrogen atom on another because they have a slight positive and negative charge. And these hydrogen bonds between the water molecules mean they're all attached in a long column. So if we looked up the stem, what we get there are lots of water molecules, all weakly bonded by these hydrogen bonds, and then as evaporation takes place, this leads to a transpiration stream up the stem. We talk right about the bottom and what's happening in the roots. What we've got here is, if we looked at the root of a plant and we took a cross section, what we'd end up with here would be kind of a cell that would look like this. In the middle, we'd have the endodermal cells. This is the endodermis here, this kind of black band. In the middle of that, this kind of star-looking shape, we've got a xylem. So here, we've got a root hair cell. And in the root hair cell, the water is being absorbed through osmosis. It moves through tissue called the cortex after passing through the epidermis here. Passes through the epidermis, through the cortex. It's going to cross the endodermis and then enter the xylem. And then it's going to move up the xylem, up the stem, and as it moves up the stem, it arrives at the leaf and this takes place. So if we just briefly talk about this process here in a bit more depth and how that water is going to enter the root hair cell, the specialization of the root hair cell, pedometer, we'll talk about that and look at the kind of experimental evidence for this. So what's going on then is, if we drew the cortex cells a bit more depth, what we've got here are cell walls. Here is a root hair cell at the end, and I'm going to draw a really big vacuole. So this vacuole is one adaptation of the root hair cell. It has this appendage which gives it a really large surface area. It has a large vacuole to give it a really negative water potential. Because it has a really negative water potential, that draws the water in. Now between each of these cells here, we've got strands of cytoplasm. And we call these strands of cytoplasm, these red bits, plasmodesmata. Now, that might be a term you've come across before or not, and this is going across a bit from the year 1 spec. Plasmodesmata is essentially just a path for water to get from one cytoplasm to another. Now that's important because there are two pathways the water can move through here. They can either go from one cytoplasm to another in something called the symplast pathway, or they can go from one cell wall to an adjacent cell wall. never actually crossing the plasma membrane. So remember, the plasma membrane is on the inside here. So it's just going through the cell wall, through the cellulose of the plant cells. We call that pathway the apoplast pathway. Now, what we're picturing here is water moving in from the root hair cell across the cortex through the apoplast and the symplast pathway until eventually they're going to reach a speed bump here. So they're going to reach this layer, the endodermis. Now, the endodermis has lots of cells. I'll draw these in a different colour so you can see the difference. The epidermis has lots of cells, plant cells that have a cell wall, but it also has a special feature in that it has a layer of a waterproof chemical called suberin. And this waterproof chemical creates a barrier. So any water that was moving through the apoplast pathway here, beg your pardon, the symplast pathway through the cytoplasm, has moved into the cytoplasm here, and then it has to cross the plasma membrane if it's going to get across the endodermis and into the xylem. Now, water that was in the apoplast pathway here, that was in the cell wall, is going to reach this waterproof barrier and it's going to be funneled into the symplast pathway. So in this way, the plant is making sure that all the water that's going to go up the plant has to cross the plasma membrane. Because remember, the plasma membrane is what controls what moves in and out of the cell. So that way, it's got a sort of a control mechanism for how much water gets up the plant. We call this waterproof layer the Casparian strip. And the Casparian strip is responsible for this control of water movement up the plant. Now, this process, this movement of the water into the xylem, then allows cohesion tension to take place, the transpiration or pull as a result of evaporation that pulls it up the plant. So, from start to finish here, if we've got more evaporation, we get more transpiration, we get more pull out the roots. But there's a few things around this. So evaporation might increase if the temperature increases. It might increase if wind speed increases. So if there's more wind speed, it blows water away and that increases the osmotic gradient. Humidity, if it increases, will decrease the osmotic gradient. So we've got lots of water outside the stomata. That means there's less of a difference and less movement of water. So those three factors would affect transpiration and pull. The size of the leaf would affect the number of stomata, so the surface area. That would be another factor. And this can all be investigated using something called a pitometer. So a pitometer is a device that allows us to measure water movement up the plant. Now, it has two kind of key features, and although it's not on the specification specifically, it's one of those ones where it's a very common experiment in biology, and it's one that we need to essentially be able to kind of understand the setup so we can explain in these kind of how science works type of questions. So what we have is, we have a plant that has been sealed using Vaseline, petroleum jelly, to create a airtight barrier. And then we have a capillary tube, a really thin tube, filled with water all the way to the end. And then we've got maybe a beaker like this filled with water, which is where all this water is coming from. Now, along this capillary tube, we've set up a ruler, and we have within the middle of it an air bubble. But what we do is, we give this various different environmental conditions. So if we were increasing the temperature, we'd put a heater next to it. If we were increasing the light, beg your pardon, I don't think we mentioned light before, did we? No, we didn't add that in. More light would mean more stomata are open, and therefore a increase in transpiration. So we might have a light, might have a heater. If we're increasing the humidity, we might put a bag over the plant to stop water evaporating and therefore increasing the humidity. All of these different environmental factors could be tested in the experiment. We have to make sure it's airtight so that the water is in one continuous column and it moves up. But how do we measure it? Well, what happens is we place an air bubble in the tube, and then we start a timer and we measure how long it takes for that air bubble to move along the tube. So if that air bubble kind of moves along. It's moving all the water, but you can get the idea. And we time how long it takes. That allows us to calculate the rate of transpiration, because we've got distance over time. Now, if we then need to reset that experiment and do a repeat, what we then need to do is move that bubble back across. So we need the bubble to get back to where it started. And that happens because of this reservoir we've got here. This will have a closed tap. And if we open the tap, that will allow water to flow through here. And because this end is sealed, that water moves in this direction and moves the air bubble back to its original position, so we can do a repeat and start again. So it has to be airtight, because if there were lots of air in the column, then that would break the chain and break the cohesive tension, and we wouldn't get an authentic, accurate measure of the rate of transpiration, because there would be pockets of air disrupting that. So the leaf itself has to be cut, or the stem has to be cut underwater to stop any air getting into the xylem tube to begin with. So it's cut underwater and then sealed underwater in the tube to prevent any air getting in the first place. So this is a pedometer, and this is what we'd use to measure that rate of transpiration as we go on.